All right, howdy. All right, so you guys might know me from your pre-lab videos. My name's Edward Lee. With us tonight is Andrew Kennedy. So he got his undergraduate degree at Providence College. He was going to go to seminary school, and I think somehow we convinced him that science was maybe the way to go. He went to graduate school at the University of Virginia and got his PhD, and then did a postdoc at the University of Alabama, Birmingham, and then started his independent career at Bates College in, what year? Yeah, we won't age it. Huh? 2016, and now he's here with us. So let's give a warm Aggie welcome to Dr. Andrew Kennedy.
All right. Can you hear me? Is my mic on? No? Hello? There we go. How about now? Is that better? All right. My name's Andrew. I'm from Bates College. This auditorium could fit the entire student body, the faculty, and the staff of my college. Today I'm going to talk to you about drugging long-term memory. And before we get started, what I'd like all of you to do is just take a few moments and recall your oldest memory from when you were very young, or at least an early memory, a happy memory. I remember mine. It was my fourth birthday, and I had just gotten a new red tricycle. I mean, it was new in a relative sense. It was my brother's Pat before that, and then I think he got it from Melissa. But it was new to me. I don't actually remember opening the tricycle. I just remember having it and having gotten it. And my first memory starts with me kind of going down the sidewalk and getting all the way to like the scary alley, and then turning around and wobbling up the sidewalk. And as I was going back towards the house, I passed the station wagon on the left. My mom drove, and she had a camera, and she was standing kind of to my right, and she took a photograph. And that's me at 4. I didn't have this photo for a really long time. When I was doing a postdoc in neurobiology studying memory, a lot of the researchers will sit around at lunch and talk about memory and what it means to us. And I was recalling this story to one of my friends, and she challenged me saying, part of the memory is your mother taking a photograph. And if that memory is real, if it's accurate, you can track down that photograph of your first memory. So I went home for Christmas, and I was working with my sister trying to go through old photo albums. We couldn't find anything that matched. My mom was a prodigious photographer. She just didn't actually develop half the film. And we eventually found this old Macy's bag full of Kodak rolls, real film. And we found one with a date that kind of matched my 4th birthday. And we developed it, and we went through it, and we found this. Oops. Memory is interesting. If you look at it from the perspective of psychology, why would I remember that experience and not others? I mean, yeah, I was scared of the alley. I was happy it was my birthday. But of course, I would have been scared or happy prior to that, and certainly since that. But I don't remember most things. But for some reason, this was salient, and I remember it. And that's interesting how we can selectively do that. From the perspective of biology, though, memory is beautiful. You have these intricate circuits within your brain. The physiology of the memory system is fascinating. Neurons get selectively activated when you experience something. They lay down these memories and give you another route to recall them. It's beautiful. However, from the perspective of chemistry, and I'm a chemist, memory is absolutely amazing. You, if you ask someone on the street 100 years ago what heredity is, they would give you a really abstract answer. They would tell you kids tend to look like their parents. However, modern genetics has fundamentally changed how we answer that question. I mean, in 1909, the word gene was invented as a theoretical construct. It wasn't like actually talking about a particular process. It was just some idea that there was something indivisible that was passed from parent to progeny that endowed this identity of heredity. And in 1945, Avery at the Rockefeller University determined that that was an Elise bacteria DNA. But now, if you ask somebody, I asked this question to my 9-year-old niece. I didn't ask her what heredity was, but I asked her, why do kids look like their parents? And what she said was genetics. And then I asked her what a gene was, and she gave me an answer that I think is absolutely astounding. She gave me the name of a molecule. I mean, it's an acronym, but a molecule. In 100 years, genetics in the 20th century turned the abstract idea of heredity into a physical thing. The revolution that is occurring in neuroscience now is very similar to the one that occurred for genetics in the 20th century. And if you ask anybody now, if I asked you, if you asked me, what is something as abstract as memory, I would give you a really fuzzy answer. But in 100 years, if you ask a 9-year-old on a playground, they're going to give you a molecular answer, maybe the name of a single molecule. The processes of the mind, things like memory, even thoughts, emotions, ideas, identity, these things are made manifest by memory. Your brain is atoms and molecules, and that is all that it is. And that means that we can use molecules to intercept these mechanisms. We can understand these mechanisms molecularly. We can erase, enhance, inhibit, and implant memories using molecules. So we're talking about cells, specifically neurons in the brain. So we have to talk about the molecules of living systems. And I promise this talk is not going to be a bunch of neuro. It's not going to be a bunch of biology terms. But we do have to talk about the central dogma of biology, the idea that many of you have seen this before, that DNA is transcribed into RNA, which is translated into protein, and it's the proteins that are the machines of the cell that make up the cell. Your neurons, the things that make the neurons work, and after all, the neurons are storing the memories in the end, they're made up of the working machines that are proteins. And we learn this central dogma in like high school biology or maybe a bio intro course in college. But nobody ever talks about how long these molecules stick around for. For example, neurons don't divide. They're postmitotic. Unlike most cells that will divide and produce progeny cells, neurons exist, and they are adults, and they do not break themselves apart. The number of neurons you have now is pretty close to the number of neurons you're going to have to use for the rest of your life. So why would a neuron even need to do this if they already have the proteins that they have? And the answer is these things break down. What is the average half-life of a protein in a cell? It's actually amazingly short. For the average protein, it's about 4 hours. Most proteins fall into a window of about 1 hour to 8 hours. Very few go beyond that. Some will last for days, but very few. So think about that. It's what, 6 o'clock, roughly? If you had a class this morning, if you had 8 a.m. lab, right? If you had 8 a.m. lab, almost all, certainly more than half of the proteins that make up your brain that are responsible for your identity and for encoding all of your memories, they're gone and have been replaced. And yet you can somehow maintain your identity and maintain memories over that span. And that's radical. So how do you do that? I mean, and first of all, what does that mean? Like, if you think about your entire body, all of these things get replaced over time. It's been calculated that over the course of about 7 years, nearly every atom in your body will be excreted in some way, shape, or form, given off, shut away, and replaced. You're still going to look kind of like you looked like 7 years ago or 7 years from now, but you'll be an entirely different organism as far as matter. That's kind of wild. When I was a kid, I remember I was camping with my family, and I was woken up by my sister in the middle of the night. We had gone to bed, and you could see storms off in the distance, but my sister woke me up because it was like extremely stormy outside. And there was this large thunderstorm and coming towards us, and my parents were like getting us out of the tent and packing things up so we could get out of there. And you could hear the sirens going off in the distance. And I was more apt to run away than help, so my sister was just like in charge of me, and I was sitting between her legs, and she was trying to preoccupy me as I looked out into the storm. And the storm was an angry thing. You could only see it when the lightning flashed, which was pretty frequent. So you get this like strobe light effect of like seeing the storm lurch across the prairie and striking out at the ground in anger. And my sister, trying to keep me calm, just said, "I learned in science class that a storm only contains the water within it for a few minutes. That it's actually absorbing humidity as it moves. It condenses that water into liquid and then sheds it. And so the actual physical stuff of the storm only exists for a few minutes." I remember hearing that and being astounded and just thinking, looking into this growling storm, like, what are you? You can look at yourself that same way. That is essentially what you're doing too. I gave a final exam in organic on Thursday. I had to push back my review session for this. My students are thrilled. But that final exam, don't tell them, the first question is labeled energy. There is nothing that freaks students out more than question number 1 on an exam being titled energy. I didn't even have a good definition for it when I was a student. Energy takes a lot of different forms, but one of them is your physical being. And we have to understand, if we're going to understand memory, how that manifests itself and how that can possibly be the case that we can maintain our identities over time as we're turning over. The key for this came from a really odd place. It came from isotope labeling in the atmosphere. So specifically carbon-14. Carbon-14 is a rare isotope of carbon. Usually carbon has 6 protons, 6 neutrons for a total atomic weight of 12. Every once in a while, at 1% of the time, it has an extra neutron, making carbon-13, and about 1 in trillion times, it will have an extra neutron still to make carbon-14. This is a level of carbon-14 in the atmosphere going back about 4,000 years. These were obtained from ice cores that were drilled in the Antarctic that capture each year the amount of carbon in the air. And so we know it's pretty consistent, the amount of carbon-14 that we had, until the 20th century. It spikes and comes back down. Like this line goes up and then back down. Let's zoom in. So you can't see it because it's off the curve, but right around 1945, this thing starts to go up, and then it really takes off in 1955. It doesn't go straight up. You look at here, there's like around 1960, there's a blip, and then it shoots up and then stops at 1963. And then from 1963 on, there's no more upward movement. The carbon-14 starts to drop in an exponential decay away from that down to this graph ends at 2010 for a reason you'll see in a second. So why would carbon-14 be stable for 4,000 years and then suddenly, in this very short period of time, go through the roof and come back down? We must have been doing something between 1945 and 1963 that was dramatically affecting the types of isotopes in the atmosphere. Talk it over. 30 seconds. What was that? All right, I hope you had some guesses. Hope you had some guesses. The answer was that humankind was throwing neutrons into the atmosphere. In 1945, over the New Mexico desert, the United States government, led by J. Oppenheimer, detonated Trinity. And when you have a plutonium or uranium fission bomb, it releases neutrons as part of the uncontrolled chain reaction. And one of the things that happens to those neutrons, besides propagating the reaction, once they get out of the core where the actual heavy uranium or plutonium exists, is they can hit nitrogen atoms in the atmosphere, which is most of the atmosphere. The nitrogen can absorb the neutron and kick out a proton. And what that creates is carbon-14. So we have nitrogen-14, neutron from the explosion comes in, producing carbon-14. It kicks out like a billiard ball, not really, but you can think of it that way, a proton. This was causing, between 1945 and 1963, this huge increase in carbon-14. It was in 1963 that Kennedy and Khrushchev signed the open-air test ban treaty that was in large part due to Linus Pauling's push to stop testing nuclear weapons just out in the open. And from that moment, the level of carbon-14 then drops down. Okay, so why do we care about this? The level of carbon-14 in the air any given year is going to be represented by the amount of carbon-14 in the plants that grew that year, and in the animals that eat those plants. So that if you were born in 1965, you were born and your body was made of plants and animals that your mother or you ate of that amount of carbon-14. Now, as you age and you start to shed atoms and replace them, you're replacing them over time with less and less enriched carbon-14. This is data from an individual who was born in 1965 and passed away in 2012. He donated his body to science, and scientists looked at different parts of the body to see at what age that body was regenerated. For example, the skin was only about 2 weeks old. That's why your cuts heal so quickly. You're constantly regenerating your skin. So the atoms that make it up are brand new to you. One part of the body was found to have this level of carbon-14 enrichment, just 5%. That corresponded to the intestine. The intestine turns over quickly, atomically. Kidneys are a little bit older. Your kidneys are going to be about 7 to 10 years old on average. But one part of the body that had a really long half-life of carbon-14 was the brain. And specifically, the scientists didn't just look at the brain tissue in bulk. They looked at the individual cells within the brain, and they looked at cortical neurons, and specifically the DNA from the neurons. They almost go back to the moment of birth of the individual. Your neurons, because they don't divide, the DNA is never copied. It's never split. It's never reproduced. And so the DNA that you have in your neurons are basically the only atoms that you carry with you for life. Everything else is being regenerated. And it's based on these data in 2005 that researchers in my group and in other groups started thinking about what's up with the DNA and could it be responsible for our ability to store information in our brains long term. This is the only neuroscience slide, I promise. It's really simple. Neurons talk to each other through synapses. When they fire together, they wire together. And for a long time, neuroscientists thought that it was these synaptic connections that were responsible for all increased and decreased connectivity between neurons throughout the central nervous system. This is true. This happens. This is called Hebbian plasticity. But there's something else that happens that's fundamental for memory. Not just at the synapse, where you have these localized mechanisms kick in to change synaptic strength, but the signal is sent all the way to the nucleus. And there are changes that occur to the genome and to the genes that are expressed off that genome when neurons are recruited for a memory experience. So we, in my lab, we focused in on these mechanisms because the DNA is there for a long time, because the gene expression that comes off the DNA regulates not synaptic plasticity, but what's called homeostatic plasticity, or really full neuron plasticity. Am I a plastic neuron or am I not? We look at these mechanisms. And they're epigenetics. If you haven't heard of epigenetics, it's okay. Epi from the Greek, which means on top of. These are biochemical reactions that happen not to the code of the DNA, but to the top of the DNA, either the proteins that it's wrapped around or directly alkylating, meaning directly reacting with the DNA. They determine if a gene that's nearby is going to be read a lot or not at all. And this is important for when talking about how your genome actually becomes functional in different cells. A neuron is like a cathedral. It is incredibly complex. You have this triangular cell body, these delicate processes like flying buttresses that come out of the center of the body, and then a long axon. If a neuron is a cathedral, this is a liver cell. A liver cell is a two-car garage with a bucket of bleach in it. It's a pretty simple cell. They have the same genome. Your liver cells have the same genome as your neurons. How does that happen? How can you give two contractors the same blueprints and one of them builds you a garage and the other one builds you a cathedral? And the reason is because epigenetics modifies the genome so that in neurons you have your neuronal genes that are being expressed. Those genes are read into proteins, that's what makes the neuron. In liver cells, the neuronal genes are silenced. And you have your liver genes that are expressed that make the liver. I kind of made fun of liver cells here, but they're actually vitally important. They're just more simple. If you didn't have your liver, you'd be dead in a couple hours. It's obviously very important to your health. But they're not the same thing as a neuron. What my lab does in general is we try to understand the biochemistry of learning and memory. What happens biochemically when you learn that facilitates memory? We then try to synthesize, using organic chemistry, small molecules that interact with those systems to try to modulate memory. Can we control it? Can we enhance it? Can we erase it? And then lastly, we design tools. I'm not going to go too much into this, but they allow us, using CRISPR, to precisely interrogate epigenetic patterns to try to recreate lived experiences. I'm going to talk about one of many epigenetic mechanisms today. It's the easiest one to understand, I think. It's called DNA methylation. A methyl group is a CH3 group. It's similar to methane, which is why it has a similar name. And methyl groups can be added to your DNA, specifically at the nucleotide cytosine. So this is cytosine. When it's in the DNA, this hydrogen is actually part of the deoxyribose. There are enzymes called DNMTs. They're DNA methyltransferases. Don't worry about that. But what they do is they can put a methyl group onto this position of the molecule so that we now have this CH3. It's called methylcytosine. This happens at about 4 to 5% of cytosines. So people in epigenetics refer to it as your fifth nucleotide. It's a separate molecule, different version of cytosine. If you look at where we see this methylation, so percent methylation from 0 to 100, it's about 4.3. This is taken from mouse hippocampal tissue. We'll get into why in a second. The hippocampus is an important part of the memory systems in mammals. However, it's really context dependent. If the next nucleotide in the DNA strand is a G, it's much more likely that that cytosine is methylated. It goes from 4% for all cytosines to 78.1 in the hippocampus. And if we consider all other contexts besides G, H just means like anything but G. It drops to basically almost 0. It's at 1.2%. This happens in what's called a CG context. Now, this also happens at both sides of the CG nucleotide. CG is really a small palindromic sequence, where the other side of the DNA is also CG going the opposite direction. And both cytosines get this methyl group attached. And what they do is they can sit in the major strand of DNA. The methyl groups are the yellow things. And they can really gunk up the ability for the DNA to be read correctly. And so because of that, these methyl marks silence gene expression. If there's a gene at that DNA, it's not going to be read if those methyl marks are there. What we wanted to do early on was simply map where methyl groups go in neurons after an animal has an experience. So we took some mice and we divided them into 3 groups. 1/3 of the mice were kept completely naive, meaning they didn't actually have an experience. They were kept in their home cage, playing with their little cardboard tube and their brothers and sisters. However, the other 2/3 had 2 different experiences. One of them was placed into a really novel context. I mean, if you're a mouse that's been hanging out with your brothers or sisters your entire life with the same cardboard tube, You know, you have some things to play with, but it's pretty consistent every single day. And then you get into a new environment, it is a big day. And so those animals will remember it. And then we had a third group that went into that same space, but then were given a mild foot shock similar to like touching a door handle and getting a static shock. It doesn't hurt, but it startles you. And they associate that startle or threat with the space. We can demonstrate this. If you put the animal back in the box, if it just saw the box, it's going to go around and play and move around a lot. But if the animal was conditioned to be afraid of the box as a threat, it will freeze, just like deer in headlights. Or really, any of you, if you're in the library and someone drops a book, quickly try to notice. There will be about 1 or 2 seconds where everybody around you is just frozen. That is a mammalian fear response. And that's what the mice do when they go back in the box. What we then did for some of the animals was 1 hour or 24 hours after training is we removed the hippocampus. The hippocampus is a part of the brain that encodes memories, especially in the first 24 hours. It's like a way station for information. It is a dense packet of neurons that are activated by experience. We divided them into regions, and we looked at 2 different things. One was what genes were being turned on and whether or not they were being methylated. I'm going to give you just one example. This is a gene that's important for plasticity. If a neuron is going to be plastic, it needs to have these pieces of RNA coming off the gene go up. You can see this outlined as the amount of RNA here coming off for both the space learning and the threat learning group. At 24 hours, because the animal is now further away from the learning experience, that RNA coming off the gene goes down. So the gene is being used really right after the experience. Well, if we look at these genes that were activated and we look at their methylation, I'm going to go through how we get these data. But what we found was that there was this increase in methylation across all the genes that were associated with plasticity. And specifically, at regions, these are called coding sequences, where the actual gene information is held. So the methyl marks were going onto the genes that were associated with plasticity. What we took away from this was the idea that this methylation process might be important for memory. So we did a lot of experiments blocking this, and what we found is if we block this process, the animals had normal cognition. They could learn just fine. They had intact short-term memory, but a completely impaired long-term memory, just of the experience that they had when we were blocking it. They could still remember older things, but we had selectively found a way to turn off long-term memory formation without affecting short-term memory formation. That's what I did right before I started my lab, and people went on to use that to try to erase memories, and they were successful, and they're investigating that as a way of trying to treat things like post-traumatic stress disorder, the way of selectively erasing a traumatic memory. I personally was more interested in enhancing memory for disease and disorders associated with memory loss. And so there's another process on this chart that I didn't mention. This methyl group that was placed on the cytosine after learning can be removed. And they can be removed by these enzymes. They're called the TET enzymes. And they oxidize or add an oxygen to the carbon of the methyl, which then eventually kicks off a pathway that replaces that cytosine and gets us back to the original starting point of normal cytosine. If DNMTs were necessary for memory formation, we hypothesized that these TET enzymes were necessary for forgetting, and we wanted to shut them down. We did this a couple ways. So TET2 was one of these TET enzymes. It has a gene, and we target it. As this gene is read, it produces messenger RNA, which will eventually go towards the protein that is TET2. What we did was we designed a complementary sequence for that messenger RNA. It's called an antisense oligonucleotide. You can think of it as similar to RNA. It hybridizes just like the double-stranded DNA would hybridize, but to the message RNA. We create this in a way that's not exactly RNA. It's actually protected, so it can last for months in a cell. And this leads to the degradation of this message RNA so that the message can never be read. So that if we look at the expression of TET2, which we're targeting, versus the other TET enzymes 1 and 3, creatively named, we can see a reduction in the TET2 gene being read out. And we can do this separately for all 3 of our target genes. So in a way, we have a way of silencing each of these genes independently. We took these ASOs and we injected them into the brain of mice. So my students that are undergrads, in fact, we did these surgeries about 2 weeks ago. One of them was done by a first year. You can do brain surgery where you drill a small hole in the skull of the mouse and then dive a needle down to the lateral ventricle. This is a sac in the brain that's important for like fluid drain out of the brain. It's basically the drain of the central nervous system. If you inject the ASO into the lateral ventricle, it can move throughout the central nervous system, knocking down your target gene anywhere it goes. So these animals had TET2 ASO injected or control into their brains. The students sewed them up, gave them some pain medication, had them recover from the surgery, and then 2 weeks later, they went and took the mice and put them into a memory experiment. This is called object location memory. The mice are put into a box that they know really, really well. But then on this day, when they go into the box, there are 2 beakers placed, these are 50 ml beakers taken from the chem lab. 2 beakers placed equidistant from this spatial cue. And the mouse will play with these 2 beakers an equal amount of time because there's nothing novel, or there's nothing differentiating the 2. 24 hours later, we can bring the mouse back into the box, and we've moved one of those beakers so that one stays in this familiar location and the other one moves to a novel location. Mice love novelty. So do you. If you were to stay in a hotel room, and it was the first time you were there, and you put your phone and keys on a table in the hotel room, and you went to bed, and you woke up and your phone had been moved, you would go and check it out. You would spend a few minutes making sure that the messages were there that you sent. The mouse is the same thing. Somebody moved its beaker and it wants to find out what's new about it. So it spends more time with the novel beaker. So we have a test for measuring the memory. If the animal remembers where the beakers used to be, they'll favor the novel location. And we call that a discrimination between the 2, and we can create this index where if the animals are favoring the novel location, they will have a positive discrimination index from 0% to 100%. And so the animals that got the control scramble ASO, they remember, but the animals that were knocking down TET2, this thing that we think is important for forgetting, they remember the experience better. We wanted to test this another way. We wanted to delete the gene. Now, you can, but it's not good to delete a gene from birth, because then the animal has to develop, and the entire brain develops without a gene. What you want to do is selectively snap your fingers and have the gene be gone. It's actually not that hard. We took TET2, and we placed 2 small sequences around an important part of the gene. These are called LOXP sites. LOXP sites are not mammalian sequences. They're only found in bacteria. And they're there to be taken advantage of by bacterial phages. These are things that infect bacteria. There's an enzyme that bacterial phages will release called Cre that binds these 2 sites and just erases everything in the middle. It cuts it out and destroys it. So that in the presence of Cre, the TET2 gene has all the important parts eliminated, and it's going to be deleted. The mouse only has LOXP sites at this gene, but it doesn't have any Cre because Cre is not a mammalian gene or a mammalian protein. We don't have this thing around. We have to artificially put it in. And so the way we put it in was we put it into the genome. We gave the mouse this bacterial phage gene, Cre. And specifically, we gave the ability for this Cre only to be expressed in genes that also express this other gene called CAMK2 alpha. These are neurons, and only neurons, that exist in the hippocampus and the frontal lobe of the mouse's brain. So that in this mouse, only those neurons, only in that part of the brain, will Cre be expressed. You can see the red part of where the Cre is being expressed here. And only there will we get a deletion of TET2. And the coolest thing is that CAMK2 alpha only turns on when the animal is about 3 weeks old. So the animal's totally normal, and then at 3 weeks, we don't do anything. In those neurons, it loses this one gene. We also had a reporter to make sure this is working. It's called a red fluorescent protein. It's also called tomato. It turns the neurons in the cortex the color of a tomato. So typical brain tissue looks like this. This is a mouse cerebellum. This is the olfactory bulb. This is the cortex on top. And You notice the cerebellum looks like normal tissue, but the cortex looks like a cherry tomato. And we could see precisely what regions of the hippocampus we were getting this gene knockout. We took these mice, we divided them in 3, did the following experiment. They all went through object location training, but then at different time points, we sent them back for testing. Some animals were tested at 2 hours. We can see the control data here, animals that didn't get CRE, and they have intact short-term memory. They can remember where the beakers were at 2 hours. Some animals were tested at 1 day. They also still remember about the same amount. But then some animals were tested at 7 days. And here's the thing. This memory is not that important. It turns out, for mice and for many of you, beakers are not that important, like their location. And I've taught a lot of organic lab and a lot of Gen Chem lab over my years. And I'll tell you that a week later, a lot of students won't remember precisely where they put their beakers. And mice can't do it either. So 7 days later, this is not actually significantly above baseline. They don't have that memory anymore. But these are the control animals. These are the knockout. At 2 hours, normal memory. Not better, not worse, just intact short-term memory. However, at 1 day and 7 days, at 1 day, the knockout animals have enhanced memory, and at 7 days, those animals can still remember where the beakers used to be. We've been able to extend this memory about 5-fold longer than neurotypical memory for animals that don't have the knockout, which is typically 3 days. So we can go about 5 times further than that. This is just a schematic of like where the animals are spending their time between the 2 different groups. Okay, we wanted to do one more test. We took the same mice and we placed them into a passive avoidance task. A passive avoidance task is where you take the mouse and you put it into a chamber that is divided light side and dark side. Mice are nocturnal, they're kind of afraid of light. So when you place them in the light side, they very quickly move over to the dark chamber where they feel more safe. But in the dark chamber, we give them that mild static shock, which gives them a little bit of a scare, and they come flying back out. We can then test them, if they remember, by placing them into the lighted chamber and measuring how long it takes them to enter back into the dark chamber. If they remember, they will take longer than the first time during the training. So here are the control animals. The first day, it took them about 15, 20 seconds to enter the dark chamber, and the next day, they are not going back in there. Makes sense. However, over time, the control animals start to reduce this latency of re-entering. Notice it never gets back down to the point that they started at during the training. It's always a little bit longer going back into the dark chamber. They're not forgetting entirely, but they're relearning. They're learning that the dark chamber is not always bad and that they can trust it a little bit, even if they take their time getting in there. Check out the knockout data. What does this mean? Like, they learn, they're afraid. On day 1 of testing, they don't go back into the box. And then they start to drop off just like the controls, but they never fully make it all the way down to that 50-second line that we see for the controls. This experiment was thought of by a student, Christine Zangler. She just graduated a PhD from UVA. She thought of it when she was a sophomore. And what these data mean is that we are not making a smarter mouse. Cognitive flexibility, which is a part of cognition, right? Part of being intelligent, is being able to take in new information and unlearn things that are no longer useful to you. As a professor, like, I know that if I say one thing that is inaccurate in class, that is the thing every student's going to remember, and it takes sometimes days to correct that. But you can, because you're smart. And this is what happens with the control mice. They figure out that the dark side is not that scary. The knockout mice don't really figure it out to the same degree. This is a deficit in cognition, but an enhancement in the original memory. And so what we think this means is that we are not making smarter mice, we're simply making smarter memories. And that is a different thing. Now, in these animals, we did the same thing, removed RNA and DNA. And we looked at expression of genes, and we looked at DNA methylation, we did this thing called whole genome bisulfite sequencing that allows us to look at the methylation status of an individual nucleotide in an individual cell. The way this works is you take bisulfite, which is a simple inorganic chemical, and you treat the DNA with it. And cytosines in the presence of bisulfite will have the bisulfite add on to it. We need to organic chemistry, you'll know that this means it broke the aromaticity of the ring. That allows for a hydrolysis of this top structure. This is called an amidine to an amide, and then eventually you get out a new nucleotide, uracil, which is an RNA. However, if the cytosine is methylated, this methyl group is big enough to sterically block the attack, and there's no reaction. So that if you treat the DNA for an hour with bisulfite, what eventually you get out is all the methylated cytosines will read out as cytosines, and all the regular cytosines read out as uracil, and we can do gene sequencing. So this is our key. A's will be read as A's, T's will be read as T's, G's are G's, but now C's are also T's. Uracil, U and T are going to be the same when being read in sequencing. Because we're reading DNA, the machine's going to think they come out as T's. And then the 5-methylcytosine will be read as a cytosine. So we're going to get a sequence off our sequencer of a piece of DNA taken from the mouse's brain, treated with bisulfite, and what we find is that there's a lot more T's than you'd expect. Moreover, if we want to find out where in the genome this thing sits, we got to scan the genome and try to find a match. This is the only cytosine that is still reading out as a cytosine. We know that blue cytosine was methylated. This is actually where in the mouse genome it came from. So we can't just control F because it doesn't match anymore. Also, the genome is 3 billion letters. So it's going to take, that's like roughly the character length of Tolstoy's War and Peace stuffed into each of your cells. So it's going to take a while to actually scan through this. And we have to simultaneously try to match it to a artificially C to D converted genome so that we can, at one point in time, find where this thing matches at least to one side of the genomes. In this way, we can map and look at genes and see like what the methylation status is of all of these different CG dinucleotides. These little ticks are methyls if they're red, or no methyl if they're blue. This is single carbon detection. This red tick means there was one cytosine that was detected with a single methyl group on it. And we can get this resolution down to within the genome wherever there's a CG dinucleotide. These gaps in the data are not gaps in the data. There's just no cytosine there that can be methylated. Quickly, because it's not that important, we're able to find that genes that get turned on after you learn something had regions that were increased methylation, exactly what we expected from the previous studies. And we see these regions, they're called differentially methylated regions, occurring at the front of the genes that are increased methylation. And so this is increased methylation associated with genes that are turned on when you learn something. So we moved on to a new idea, which was, if we can make a mouse that has better memory, can we make a mouse that selectively, one memory is stronger than other memories within the same mouse? And so we took a different mouse that still has TET2 with those LOXP sites in it, but we used a different driver of Cre. We used FOS, which is a gene that's only turned on when neurons are activated. And we hooked up the Cre to it, and we gated it by an estrogen receptor, which allows us to use an estrogen antagonist, which is a really weird thing, don't get me wrong, to turn this Cre on. When you learn something, not all of your neurons in your brain get activated. A very small amount. Depending on the experience, anywhere between 1% to maybe upwards of 10% above baseline. When I was a kid, I don't know if people still say this, when I was a kid, I would hear people say, you only use 10% of your brain. Imagine how smart you would be if you used 100% of your brain. Has anyone ever heard that? People still say that? Okay. You can use 100% of your brain. You can have all of your neurons fire at once. It's called a grand mal seizure. There is a reason you only use about 10% of your neurons at any given time. It's a bit like saying Chopin's music is incredibly powerful and emotional and beautiful. But did you know he only hits about 5 keys at a time? Imagine how amazing Chopin's music would be if you hit all 88 together. That's not how your brain functions. And so when you learn something, it's really a small number of neurons that are going to be recruited for that memory. And that's good. It leaves more space for other experiences to be encoded. But those neurons that are in this mouse have Cre expressed, and then they can knock out TET2 only if we inject the mouse with tamoxifen directly before. So memory 1, mouse gets either tamoxifen or vehicle. 2 hours later, they go into the box with the beakers. 7 days later, they go back and they get tested. We wait a month. Same mice go through a second memory. We don't use beakers this time, we use Legos, which I don't understand this at all, mice. They like Legos as much as they like beakers. They get either tamoxifen or vehicle. Here's the thing, if they got tamoxifen the first memory, they get vehicle the second. If they got vehicle the first memory, they get tamoxifen the second. So one of them, only one of them is labeled with tamoxifen, and only one of those networks of neurons get the knockout. 7 days is a time point where neurotypical mice can't remember. If the animals get the tamoxifen, that memory, they can. These lines are the data from connecting the same mouse, when it got the tamoxifen or when it didn't. And then these are the control mice. Notice that unless they have the Cre and the tamoxifen for that memory, they don't remember. That means this mouse has one set of neurons with one memory that is stronger than another set of neurons for another memory. So we wanted to not just do gene knockouts, we wanted to make an organic molecule that could be translatable to the clinic to be useful as a drug. And so there was no TET inhibitors. These didn't exist. The enzymes had been discovered only about 2 years before this. But there was a crystal structure, meaning we had a crystal structure is just a way of getting a 3D structure of a molecule, including proteins, although it's hard. There was a structure of what TET2 looked like when it was bound to double-stranded DNA that had been methylated, so like methylated DNA. And if you zoom in, you could see this 5-methylcytosine, this methylcytosine in the active site of this TET enzyme about to oxidize it and about to remove that methyl mark. We can even have a 2D representation of all the different parts of the protein, the amino acids that make it up, and how they're coming in contact with the methylcytosine. We use this information to design a molecule to mimic the methylcytosine. What we did was we took cytosine, it's cheap, made by nature, and we added a chlorine to the position where the methyl used to be. Chlorines are nothing like a methyl, they're not tetrahedral, they're just spherical, but they're about the same size. So in medicinal chemistry, if you want to mimic a methyl that's about to react in some way, you can use a chlorine, because that methyl might be oxidized by the TET enzymes, but a chlorine won't. We then replaced the deoxyribose of the DNA with rings that couldn't be reacted in the same way that a ribose is reacted. The first thing we did was we varied what this could be at this position. We tried hydrogen, chlorine, bromine. We even tried a trifluorocarbon. The chlorine worked the best, as we thought it was going to work the best, and it did. This assay is just the percent inhibition we're getting at a certain concentration, 100 micromolar of the inhibitor. We then varied what this ring was at the bottom. We tried a lot of things. We found this one here, this 3-biphenyl. I'll show you what it looks like. So you have the cytosine right in the center, a chlorine where the methyl used to be, and then these 2 rings off of it, and it inhibits the TET enzymes decently well. And so we thought we'd take this thing and see if we could increase methylation in neurons. So we threw it on neurons. We took mouse hippocampal neurons, and we added in 2 of our compounds that were working, and we found was that the product of the enzymatic reaction was dropping. And the substrate, the methylcytosine, was increasing. Notice, this is not a lot. We're increasing by 0.5% over the genome. But remember, the methylation is important for encoding if the cell is a neuron or a liver cell. We're not going to be shifting huge amounts of methyl groups. We're going to be changing a small amount of them. The idea being that if a huge change in methylation is cell identity, a small change might be cell personality, and that you can convert a neuron from being plastic to aplastic or vice versa. I'm not going to go through all of this, but a lot of them were hypermethylated, increased methylation at particular genes, specifically pathways in cancer and also glutamatergic synapses. Pathways in cancer are also utilized for plasticity in neurons. Just like cancer cells start to grow wildly, neurons use those same genes to grow wildly when they make new connections. They just can't divide, so they can't become cancerous very easily. We took mice. We trained them in a box with beakers. We then gave them an injection of this inhibitor called Bobcat 339. My school, the mascot is Bobcats, and I let the students name the molecules. So inhibitor or vehicle, 22 hours later, so 1 day from training, they then got tested. We get penetration into the brain for a couple hours in the range where you see inhibition. This is not a long time. A half-life of a couple hours for a drug is not ideal. Typically, you want 8 hours. It's an ideal half-life for a drug. That way you can take it once a day and keep boosting levels back up to where you want them. 2 hours is short. But yet, even with that, the animals that got treated with the inhibitor had enhanced memory at 24 hours. Just so you know how this can also translate into other people's research, like we make these tools and we utilize them, but other people can pick them up. Like you can buy this compound on Aldrich, which is a chemical company. And this was published 3 days ago in the Proceedings of the National Academy of Sciences. A team at Yale picked up Bobcat 339, our inhibitor, and they found that not only was it inhibiting the enzyme, but it was also degrading the target protein. By binding to the enzyme, it was causing the protein to basically fall apart and be destroyed by the proteasome. So that not only was it inhibiting the enzymatic activity, but it was eliminating the protein from the cell, and specifically in neurons. So here is the neurons. They were interested in the hypothalamus because they were studying feeding behavior. They think this could be useful for treating anorexia. And in the vehicle, the TET3 protein is being labeled in these neurons, you can see here, as red. And when they're given the Bobcat 339, we aren't just inhibiting, but the red is being eliminated. We're actually removing TET3 from the cells entirely. So there is no protein to do the chemistry anymore. And what they found was they took these animals through a regimen of first giving them a wheel to play with, so they had some exercise, and then they had food ad lib, which means that they had food 24 hours a day, which is pretty normal for a mouse. At P40, which is 40 days after birth, they gave either the inhibitor of the TET enzymes we had made or vehicle, and then they started a food restriction assay, which is supposed to mimic anorexia. It's in a mouse, it's not the same thing, but it does cause anxiety, and it does cause body weight reduction because they simply eat less. They could eat, they have 2 hours to eat, but they don't because they're anxious about it. And if they had vehicle, they lose weight, and this is typical. But the animals that had TET inhibition managed to maintain that weight, and you can see they're eating more during the food-restricted period. So what we think is happening for both our memory experiments and for their feeding behavior experiments, which is driven by the hypothalamus, is that these epigenetic changes are governing neuronal function. And because the inhibitor is both inhibiting the enzyme when it's there, but also degrading the enzyme, it can lead to a long-lasting and immediate effect for the drug. So we're pretty excited about that. Conclusions from just our work in our lab: the TET enzymes do appear to be negative regulators of memory. They're forgetting genes. We can target them, we can delete them, and we can inhibit them. In all those cases, we can enhance memory. This work is entirely done by undergraduates. Every last experiment, every surgery. The synthesis was done of the molecules done by this team. This is them at ACS a couple years ago. These are my neuroscientists at Society for Neuroscience. This is my current lab. We just went out to dinner a few months ago. And then these are my funding agencies, which include the Penn Medicine Orphan Disease Center. One thing I didn't talk about was we're trying to target specific types of autism that have memory deficits. The Pitt Hopkins Research Foundation, the state of Maine, but also the National Institutes of Health, both the General Medicine and the Mental Health Institutes. All right, everybody, thanks for coming. If you have any questions, please come up and talk to me afterwards if you want. I'm taking a selfie.